You know that feeling maybe a few hours after lunch when your energy just dips? Or, thinking bigger picture, the complex metabolic dance people managing diabetes navigate every day.
Right. It all comes down to fuel, how our bodies make energy.
Exactly. And what happens when glucose, our go-to fuel, isn't readily available? Well, it turns out our bodies have these really sophisticated backup plans.
Absolutely. The body doesn't just grind to a halt. No, it cleverly switches gears, tapping into stored fat. It's a fascinating, intricate biochemical process.
And that's what we're doing today, a real deep dive. We're not just skimming the surface.
No, forget the high-level overview.
We want you to come away with a mastery of this, like the professionals in the field. We're going deep into how fats are broken down for energy, that's fatty acid oxidation, and also how the body makes ketone bodies.
An alternative fuel source.
Precisely. So our mission today is crystal clear. We're going to unpack the detailed biochemistry, drawing on our sources, leaving no stone unturned.
We want a complete, nuanced understanding of fatty acid oxidation, ketogenesis, how they're controlled, and why they matter, in health and disease.
Okay, let's jump in. Fatty acid oxidation, the main engine for using fat. Where does this happen? Inside the cell, specifically?
Primarily, yeah, it happens inside the mitochondria, the cell's powerhouses.
Right.
And this location is really key because the process of making fatty acid synthesis happens out in the cytosol, a completely different compartment.
Ah, so they're kept separate.
Exactly. This separation is crucial for control. It prevents the body from, breaking down fat and making it at the same time, which would be incredibly wasteful. It allows the body to integrate energy production with what the tissues actually need.
So breaking down fat isn't just running the synthesis pathway backward.
Not at all. It's a distinct pathway. Fatty acid catabolism, the breakdown, uses different enzymes, requires fatty acids to be in an activated form called acetyl-CoA.
Okay.
It uses specific coenzymes, NAD+, and FAD. It generates ATP, and critically, it needs oxygen. It's an aerobic process.
Okay, aerobic. Got it. Now, these fatty acids, especially the longer ones, they're fats. How do they even get around in our watery blood to the cells?
Good question. They aren't really free, longer chain ones, sometimes called FFAs or NEFAs.
Right, free fatty acids.
Yeah, well, they actually bind to albumin in the blood plasma. Albumin acts like a taxi service for them.
Okay.
Then, once inside the cell, they often bind to other proteins, fatty acid binding proteins, to move around. Shorter chain fatty acids are a bit more water soluble, so they can travel more easily, sometimes as ions.
All right, so the fatty acid arrives at the cell. What's the very next step before it can be broken down in the mitochondria?
Activation. You have to activate the fatty acid first. Think of it like priming it.
And this costs energy.
Yes, this is actually the only step in the whole breakdown process that uses ATP directly. An enzyme called acetyl-CoA synthetase, or biokinase, does the job. It uses ATP and coenzyme A-CoA to attach the CoA molecule to the fatty acid, forming acetyl-CoA.
Okay, so that's the activated form.
Precisely. And this reaction produces AMP and pyrophosphate, PPI.
Pyrophosphate, PPI. Does the cell just let that build up?
No, good point. There's another enzyme, inorganic pyrophosphatase, that quickly breaks down that PPI into two inorganic phosphates.
Oh, okay.
And breaking that bond releases more energy, which basically makes the initial activation step irreversible. It pulls the reaction forward.
Clever. Where are these activating enzymes, these acetyl-CoA synthetases, located?
They're actually found in a few places, on the outer mitochondrial membrane, which makes sense,
Right.
but also on the endoplasmic reticulum and even in peroxisomes, different locations for potentially different roles.
Okay, so the long-chain fatty acid is now activated to acetyl-CoA, but it's still outside the inner mitochondrial membrane where the main action happens. How does it cross that barrier?
Right, that inner membrane is pretty impermeable to long-chain acetyl-CoA. This is where carnitine comes in. It's a small molecule, very abundant in muscle.
Carnitine. I've heard of that.
Yeah, it's absolutely essential. There's an enzyme on the outer mitochondrial membrane called CPTI, carnitine palmitoyl transferase -I. It acts like a gatekeeper. CPTI.
CPTI takes the acyl group from acetyl-CoA and transfers it to carnitine, forming acylcarnitine and releasing the CoA back into the cytosol.
So it swaps the CoA tag for a carnitine tag.
Exactly. Kind of like a temporary passport to get across the border. The acylcarnitine can then move across the inner membrane.
How does it do that?
It uses a specific transporter protein called the carnitine acylcarnitine translocase.
Right.
Think of it like a revolving door. It swaps one acylcarnitine going into the matrix for one free carnitine coming back out.
Okay, efficient.
Very.
Mm-hmm.
Then, once inside the matrix, another enzyme, CPT2, carnitine palmitoyl transferase 2, which sits on the inner side of the inner membrane, reverses the process.
Swaps it back.
It swaps back. It transfers the acyl group from carnitine back to a CoA molecule that's already inside the matrix, reforming acetyl-CoA there. And the free carnitine gets sent back out by the translocase to pick up another passenger. It's a really elegant shuttle system. It really is. Okay, so now, finally, the activated fatty acid, acetyl-CoA, is inside the mitochondrial matrix. Let's get into the details of via oxidation itself. How does it chop off those 2 carbon units?
Right, via oxidation. It's a cyclical process that sequentially removes 2 carbon units as acetyl-CoA.
Acetyl-CoA, the molecule that can feed into the citric acid cycle.
Exactly. The breakdown starts at the carboxyl end of the fatty acid chain, and the key cleavage happens between the second carbon, the alpha carbon, and the third carbon, the beta carbon, hence the name via oxidation. So for example, take palmitoyl-CoA, which has 16 carbons. It goes through the cycle 7 times.
7 cycles, releasing
8 molecules of acetyl-CoA in total. 7 cleavages give you 8 pieces.
Got it. And each cycle involves a set sequence of reactions.
Yes. 4 core steps per cycle, repeated over and over. Step 1 is dehydrogenation. An enzyme called acetyl-CoA dehydrogenase removes hydrogens from the alpha and beta carbons.
Okay, dehydrogenation. Does that involve those coenzymes you mentioned?
It does. This first step requires FAD. It takes the hydrogens, becoming FADH2, and creates a double bond in the fatty acid chain, forming trans-anoyl-CoA. So right away, we're generating an energy carrier, FADH2.
FADH2, okay. First energy payout, what's step 2?
Step 2 is hydration. Another enzyme, anoyl-CoA hydratase, adds water across that double bond.
Saturating it again.
Exactly. Saturating the bond and forming 3-hydroxyacyl-CoA.
Okay. Then step 3.
Step 3 is another dehydrogenation. This time the enzyme is L3-hydroxyacyl-CoA dehydrogenase. It removes hydrogens from that hydroxyl group on the third carbon.
And this uses NAD+.
Yes. This step uses NAD+ as the coenzyme, which gets reduced to NADH, another energy carrier generated. And the product is 3-ketoacidyl-CoA.
So FADH2 from step 1, NADH from step 3. What's the final step, the cleavage?
The final step, step 4, is thiolytic cleavage. An enzyme called thiolase, or 3-ketoacyl-CoA thiolase, uses another molecule of CoA to break the bond between the second and third carbons of the 3-ketoacyl-CoA.
Splitting it off.
Right. This releases 1 molecule of acetyl-CoA, that's our 2-carbon unit, and leaves behind an acyl-CoA molecule that's now 2 carbons shorter.
And that shorter acetyl-CoA just goes back to step 1.
Exactly. It re-enters the cycle, goes through the 4 steps again, gets shorter, releases another acetyl-CoA, and so on, until the whole chain is broken down. For even chain fatty acids, anyway.
Right. What about odd-chain fatty acids? You mentioned them briefly. You can't end up with acetyl-CoA at the end, can you?
You're right. For odd-chain fatty acids, oxidation proceeds normally, chopping off acetyl-CoA units, until you're left with a final 3-carbon fragment. That's propionyl-CoA.
Propionyl-CoA, what happens to that?
It doesn't go through oxidation. Instead, it enters a separate short pathway. It gets carboxylated, then rearranged, and ultimately converted into succinyl-CoA.
Succinyl-CoA. That's an intermediate in the citric acid cycle, isn't it?
It is, and that's a key point. Because succinyl-CoA can be converted eventually to glucose precursors, this means that the 3-carbon tail of odd-chain fatty acids is glucogenic. It can be used to make glucose.
Unlike the acetyl-CoA from even-chain fats, which generally can't be used to make net glucose.
Precisely. That's a really important distinction.
Okay, so back to the energy yield. We're generating FADH2 and NADH in each cycle. How does that translate into ATP?
Right, so each cycle gives 1 FADH2 and 1 NADH. Let's use palmitate, our 16-carbon example again. 7 cycles give 7 FADH2 and 7 NADH.
Okay.
These electron carriers, FADH2 and NADH, then shuttle their high-energy electrons to the electron transport chain, which is embedded in that inner mitochondrial membrane.
Right, oxidative phosphorylation.
Exactly. As electrons move down the chain, protons are pumped out, creating a gradient, and the flow of protons back in through ATP synthase drives ATP production.
And how much ATP do we get per carrier, roughly?
Generally, oxidizing 1 FADH2 yields about 1.5 ATP, and 1 NADH yields about 2.5 ATP. So from the 7 FADH2 and 7 NADH from palmitate oxidation.
That's 7 times 1.5, which is 10.5 ATP, plus 7 times 2.5, which is 17.5 ATP. So 28 ATP just from the oxidation cycles themselves.
Correct, just from the electron carriers produced during the cycles. But don't forget the acetyl-CoA.
Yes, we got 8 molecules of acetyl-CoA from palmitate.
Right, and each of those acetyl-CoA molecules enters the citric acid cycle also in the mitochondrial matrix.
And the citric acid cycle generates more NADH, FADH2, and some GTP.
Exactly. Each turn of the citric acid cycle, oxidizing one acetyl-CoA, yields roughly 10 ATP equivalents when you factor in the oxidative phosphorylation of its products.
So 8 acetyl-CoA times 10 ATP each. That's another 80 ATP.
That's right. So if we add the 28 ATP from the oxidation cycles and the 80 ATP from the acetyl-CoA oxidation,
We get 108 ATP. That seems like a lot.
it is. But wait, there's one small adjustment.
The activation step. We used ATP at the very beginning.
Exactly. That initial activation of palmitate to palmitoyl-CoA costs the equivalent of 2 ATP molecules. Remember, it produced AMP and PPI. That's like spending 2 high-energy bonds,
Okay, so we subtract 2 ATP from the 108.
giving us a net yield of 106 ATP per molecule of palmitate.
Wow, 106 ATP. Compare that to glucose breakdown.
It's much, much higher per carbon atom. This really shows why fat is such an incredibly efficient way to store energy. We capture about 1/3 of the total energy potential in palmitate as ATP.
Incredible efficiency. Now, you mentioned peroxisomes earlier, involved with very long-chain fatty acids. How is oxidation different there?
Right. Peroxisomes handle the really long chains, say C20, C22, and some branched ones. They have a modified oxidation pathway.
Modified how?
The main difference is the first step, the initial dehydrogenation. In peroxisomes, the enzyme, acetyl-CoA oxidase, transfers electrons directly to oxygen O2 instead of FAD.
To oxygen, what does that produce?
It produces hydrogen peroxide, H2O2.
Hydrogen peroxide. Isn't that harmful?
It can be, but peroxisomes are packed with another enzyme called catalase, which immediately breaks down the H2O2 into water and oxygen. Very neat cleanup.
Okay. And does this peroxithermal oxidation make ATP directly?
No, that's another key difference. It's not directly linked to ATP synthesis like mitochondrial oxidation is. The main job seems to be shortening those very long chains.
So they shorten them, and then what happens?
The shorter fatty acids, often as acetyl-CoA or medium-chain acyl-CoAs, are then transported to the mitochondria for complete oxidation there. So peroxisomes do the initial trimming.
Got it. Any other roles for peroxisomal oxidation?
Yes, it's also involved in making bile acids, synthesizing certain types of lipids called ether glycerolipids, and steps in cholesterol synthesis. And interestingly, high-fat diets or certain drugs can actually increase the activity of these peroxisomal enzymes.
Fascinating. Okay, what about unsaturated fatty acids, the ones with double bonds? How does oxidation handle those kinks in the chain?
Good question. It mostly uses the same enzymes, but it needs a couple of extra helper enzymes to deal with the double bonds when they end up in awkward positions.
Awkward positions.
Yeah. The standard enzymes work fine until a double bond is encountered at, say, the delta 3 position. A normal intermediate has the double bond at delta 2.
So what happens at delta 3?
An enzyme called an isomerase just shifts the double bond from the delta 3 position to the delta 2 position. It converts it into the standard trans delta 2 intermediate, and then acidation can just continue as normal.
Okay, so an isomerase fixes that. What if the double bond is at delta 4?
That's a bit trickier. That requires 2 extra steps. First, a reductase enzyme uses NADPH to reduce the double bond system.
NADPH, where did that come from inside the mitochondria?
From other mitochondrial reactions, like those catalyzed by glutamate dehydrogenase or isosocitrate dehydrogenase.
Okay, so reductase acts first. Then what?
After the reduction, you end up with a double bond at the delta 3 position, which is then handled by that same isomerase we just talked about. It shifts it to delta 2 and boom, back on the main pathway.
So a combination of reductase and isomerase allows the pathway to navigate those double bonds.
Exactly. It's a clever set of workarounds to make sure unsaturated fats can also be fully oxidized.
Really shows the pathway's adaptability.
Okay, let's switch gears slightly. When fatty acid oxidation is running really high, especially in the liver, another process kicks in. Ketogenesis, making ketone bodies.
That's right. Under conditions like prolonged fasting, very low-carb diets, or uncontrolled diabetes, the liver ramps up fatty acid breakdown so much that it starts producing significant amounts of these ketone bodies.
And these are
Acetoacetate, D3-hydroxybutyrate, sometimes called beta-hydroxybutyrate, and acetone.
acetone, like nail polish remover.
Chemically, yes. Acetoacetate is unstable and can spontaneously break down into acetone and CO2. Acetone is mostly just breathed out.
Hence the fruity breath smell, sometimes associated with ketosis.
Exactly. The main players metabolically are acetoacetate and D3-hydroxybutyrate. And technically calling them all ketones isn't quite right biochemically, but the term ketone bodies is standard.
Can acetoacetate and D3-hydroxybutyrate be converted into each other? Yes, easily. There's a mitochondrial enzyme, D3-hydroxybutyrate dehydrogenase, that interconverts them. The balance depends on the NDD plus NADH ratio in the mitochondria, the redox state.
Okay. And normally, are ketone body levels high?
No, usually very low in well-fed mammals, less than a zero point 0.2 millimolar. Ruminants are a bit different. They make some continuously from gut fermentation products. But for us, the liver is the main source when levels rise.
So the liver makes them, but other tissues use them.
Precisely. Tissues like muscle, heart, and importantly, the brain, especially during fasting, can take up acetoacetate and 3-hydroxybutyrate from the blood and use them as fuel.
The brain, too. I thought the brain primarily used glucose.
It does, primarily. But during prolonged fasting, when glucose is scarce, the brain adapts remarkably well to using ketone bodies for a significant portion of its energy needs. Acetone, as we said, is mostly waste, so there's a net flow. Liver produces, other tissues consume.
What's the starting material for making them in the liver mitochondria?
It starts with acetyl-CoA, the end product of beta-oxidation. 2 molecules of acetyl-CoA condense together.
The reverse of the last step of oxidation.
Essentially, yes. Catalyzed by the thiolase enzyme, forming acetoacetyl-CoA.
Okay, acetoacetyl-CoA. Then what?
That acetoacetyl-CoA then reacts with another molecule of acetyl-CoA. This is catalyzed by HMG-CoA synthase.
HMG-CoA synthase.
Forming
HMG-CoA, which stands for 3-hydroxy-3-methylglutarol-CoA.
Right, HMG-CoA. I know that's also an intermediate in cholesterol synthesis.
It is, but the HMG-CoA synthase involved in ketogenesis is specifically in the mitochondria, whereas the one for cholesterol synthesis is in the cytosol. Different pools, different fates.
Ah, okay. So mitochondrial HMG-CoA is for ketone bodies. What happens next?
Another mitochondrial enzyme, HMG-CoA lyase, then cleaves that HMG-CoA. It splits off a molecule of acetyl-CoA, leaving free acetoacetate.
And that's our first main ketone body.
That's it. And then, as we mentioned, acetoacetate can be reduced to D3-hydroxybutyrate using NADH, catalyzed by D3-hydroxybutyrate dehydrogenase. Often, 3-hydroxybutyrate becomes the more abundant ketone body in the blood during ketosis. So, the liver makes these ketone bodies, but doesn't seem to use much of them itself. How do the other tissues, like muscle and brain, actually burn them for energy?
That's a key point. The liver lacks significant amounts of a crucial enzyme needed to reactivate acetoacetate back into acetoacetyl-CoA effectively within the mitochondria.
Which enzyme is that?
It's often called CoA transferase, or more formally, acetyl-CoA acetoacetate-CoA transferase. Extrahepatic tissues have this enzyme.
Oh, what does it do?
It takes the CoA group from acetyl-CoA, an intermediate from the citric acid cycle, and transfers it onto acetoacetate, reforming acetoacetyl-CoA inside the mitochondria of those user tissues.
Okay, so it reactivates it using a CoA from the citric acid cycle. Clever?
Very. Once you have acetoacetyl-CoA again, thiolase just splits it into 2 molecules of acetyl-CoA.
And those acetyl-CoA molecules just enter the citric acid cycle.
Exactly. They get oxidized just like acetyl-CoA from glucose or fatty acid breakdown, generating ATP.
What about the D3-hydroxybutyrate?
That gets oxidized back to acetoacetate first, using that D3-hydroxybutyrate dehydrogenase enzyme again. This step actually produces an NADH molecule, giving a bit of extra energy. Then the acetoacetate is activated and split as we just described.
So using ketone bodies yields a good amount of ATP too.
Yes. Acetoacetate yields about 19 ATP, and 3-hydroxybutyrate yields around 21.5 ATP. When ketone body levels get really high, say around 12 millimolar, they can actually saturate the oxidative machinery in tissues like the brain, contributing a huge fraction of energy production.
What about getting rid of them? You mentioned acetone via the breath. Can they also be lost in urine?
Yes. When blood levels, ketonemia, are only moderately high, the kidneys can reabsorb most of them, so urinary loss, ketonuria, is minor.
But there's a limit.
There is. It's not a super sharp threshold, it varies. But once blood levels get high enough, the kidney's reabsorption capacity is overwhelmed and ketone bodies start spilling into the urine. That's ketonuria.
So measuring blood levels is probably a better indicator of how severe the ketosis is.
Generally, yes. Ketonemia gives a more direct measure than just finding ketones in the urine.
Okay, this brings us nicely to regulation. How does the body control this whole process, making sure ketogenesis doesn't run wild?
Regulation is critical, and it happens at basically 3 key levels. The first control point is getting the fuel in the first place. mobilizing free fatty acids from fat stores.
From adipose tissue.
Exactly. You won't get significant ketosis unless you have high levels of FFAs circulating in the blood, released from adipose tissue via lipolysis. The liver is very good at grabbing these FFAs. It takes up about 30% of whatever flows through it.
So if blood FFA levels are high, liver uptake is high.
Right. Therefore, anything that controls lipolysis in fat tissue hormones, like insulin and glucagon, for instance, is fundamentally controlling the supply of raw material for ketogenesis. That's checkpoint number 1.
Okay, supply control. What's the second checkpoint?
The second point is within the liver itself. Once the fatty acids get into the liver cell, they face a choice. Either get oxidized in the mitochondria, leading to ATP and maybe ketone bodies, or get repackaged into triglycerides and phospholipids, often for export as VLDL.
Oxidation versus esterification.
Precisely. And the key gatekeeper here is that enzyme we met earlier, CPTI, carnitine palmitoyltransferase I. It controls entry into the mitochondria for oxidation.
Right, the shuttle system's entry point.
Exactly. In the well-fed state, CPTI activity is relatively low. Fatty acids coming into the liver tend to be esterified. During fasting or low-carb states, CPTI activity goes up, favoring oxidation.
What controls CPTI activity?
A really important regulator is malonyl-CoA.
The first molecule committed to making fatty acids.
That's the one. Malonyl-CoA strongly inhibits CPTI. So when you're well-fed and making fat, high malonyl-CoA, you inhibit CPTI, preventing the fat you just made from immediately being burned. Makes sense, right?
Yeah, prevents a futile cycle.
Exactly. But during starvation, FFA levels rise, insulin drops, glucagon rises. These conditions lead to lower malonyl-CoA levels inside the liver cell.
How do lower malonyl-CoA levels happen?
Rising fatty acyl-CoAs inhibit the enzyme that makes malonyl-CoA, acetyl-CoA carboxylase, and the hormonal changes also favor its inactivation. So lower malonyl-CoA means less inhibition of CPTI.
Releasing the brake on CPTI.
Right, allowing more fatty acids into the mitochondria for bioxidation. So CPTI is a critical switch point, deciding between burning fat and storing exporting it.
Okay, so control point 1 is FFA supply. Control point 2 is CPTI regulating entry into mitochondrial oxidation. What's the third?
The third point involves what happens to the acetyl-CoA after it's produced by bioxidation inside the mitochondria. It can either enter the citric acid cycle for complete oxidation, or it can be diverted into the ketogenesis pathway.
So citric acid cycle versus making ketone bodies.
Yes. And what's interesting is that as the rate of fatty acid oxidation increases, meaning more acetyl-CoA is being produced, a larger proportion of that acetyl-CoA gets channeled into ketone body production.
That seems counterintuitive. Why not just burn it all in the citric acid cycle if fuel is available?
It seems to be partly about maintaining energy balance within the liver itself. Remember, making ketone bodies yields less ATP for the liver compared to completely oxidizing the acetyl-CoA. By shunting some acetyl-CoA to ketone bodies for export, the liver can process large amounts of fatty acids without necessarily overwhelming its own capacity for oxidative phosphorylation. It's like an overflow valve.
An overflow valve. Interesting. Is there another reason acetyl-CoA might be diverted from the citric acid cycle?
Yes, a very important one. The availability of oxaloacetate.
The molecule acetyl-CoA combines with to enter the citric acid cycle.
Exactly. If oxaloacetate levels in the mitochondria drop, the citric acid cycle slows down simply because there isn't enough oxaloacetate to combine with the incoming acetyl-CoA.
And why might oxaloacetate levels drop?
Two main reasons in states favoring ketosis. First, high rates of oxidation produce a lot of NADH. This high NADH-NAD plus ratio pushes the equilibrium of an enzyme, malate dehydrogenase, away from oxaloacetate towards malate. Second, during fasting or low-carb states, the liver is often also performing gluconeogenesis, making glucose.
And gluconeogenesis uses oxaloacetate as a starting material.
It does. So oxaloacetate is being consumed for glucose synthesis, further lowering its availability for the citric acid cycle. The result is that acetyl-CoA builds up and gets diverted more strongly towards ketogenesis. While there are mechanisms to try and replenish oxaloacetate in severe starvation or uncontrolled diabetes, they get overwhelmed.
Wow, that's a really intricate interplay between fat oxidation, the citric acid cycle, gluconeogenesis, and ketogenesis, all regulated by substrate availability and redox state.
It truly is. The liver is performing an incredible balancing act.
Which leads us to what happens when this balancing act fails. What are some clinical consequences if fatty acid oxidation is impaired?
Right. If you can't oxidize fats properly, you often run into problems, especially hypoglycemia, because you lose that key alternative fuel when glucose is low. Carnitine deficiency is one example.
Carnitine deficiency, how does that happen?
It can occur in newborns, especially preemies, maybe due to poor synthesis or kidney losses. People on hemodialysis can lose carnitine. Some might even need it as a dietary supplement.
And the symptoms.
Typically hypoglycemia, but without high ketone bodies because you can't get the fats into the mitochondria properly. Also, fat accumulation in muscles leading to weakness. Oral carnitine can often treat it.
Are there inherited defects in the system?
Oh, definitely. Inherited defects in CPT mostly affect the liver, causing hypoglycemia during fasting because fat oxidation and ketogenesis are
The
reduced.
CPT2.
CPT2 deficiency is more common and usually affects muscle, causing pain and weakness with exercise or fasting. Severe forms can affect the liver too. Interestingly, some diabetes drugs, sulfonylureas like glyburide, actually inhibit CPTI, which contributes to their glucose-lowering effect by reducing fat oxidation.
What about defects in the oxidation enzymes themselves?
Yes, inherited defects exist for pretty much all the enzymes in the pathway. Things like long-chain or short-chain 3-hydroxyacetyl-CoA dehydrogenase deficiency. The long-chain one is linked to a serious condition called acute fatty liver of pregnancy. Also, deficiencies in thiolase or HMG-CoA lyase, these typically present with non-ketotic hypoglycemia, coma, and fatty liver.
Are there any conditions caused by toxins affecting fat oxidation?
A classic example is Jamaican vomiting sickness. It's caused by a toxin, hypoglycin, in unripe aquihu fruit.
What does hypoglycin do?
It inactivates medium and short-chain acetyl-CoA dehydrogenases, basically shutting down oxidation and causing severe hypoglycemia. Very dangerous.
Any others?
There's dicarboxylic aciduria, often caused by MTAD deficiency, medium-chain acetyl-CoA dehydrogenase deficiency. You see medium-chain dicarboxylic acids in the urine and non-ketotic hypoglycemia. Refsum disease is a neurological disorder from a defect in oxidizing phytanic acid, a branched-chain fat found in dairy and ruminant meat. Its buildup causes nerve problems. And Zellweger syndrome is a devastating condition where peroxisomes are absent or non-functional, leading to accumulation of very long-chain fatty acids and severe neurological issues because that initial trimming step can't happen.
So, problems if fat oxidation is too low. What about the flip side? Ketoacidosis from too much ketone production.
Exactly. High levels in blood, ketonemia, or urine, ketonuria, define ketosis. A mild form happens in starvation, which is usually okay.
But it can become pathological.
Yes, severe dangerous ketosis can occur in uncontrolled type 1 diabetes or certain conditions in animals like twin lamb disease or ketosis in dairy cows. Even high-fat diets or severe exercise can cause milder, non-pathological ketosis.
And why is it called ketoacidosis? Where does the acid come from?
Acetoacetic acid and D3-hydroxybutyric acid are, well, acids, moderately strong ones. Normally, the body's buffering systems, like bicarbonate in the blood, handle the small amounts produced.
But if production skyrockets?
If production is excessive, these acids overwhelm the buffering capacity. The body keeps losing bicarbonate as it tries to buffer and excrete the acids. Blood pH starts to drop significantly.
Leading to acidosis.
Metabolic acidosis, yes. And in uncontrolled diabetes, this ketoacidosis can become extremely severe and life-threatening if not treated urgently.
This has been incredibly thorough. We've gone really deep into the mechanics and regulation of fat metabolism. To wrap up, what are the absolute key takeaways for mastering this topic?
Okay, key points. 1, fatty acid oxidation in mitochondria via Bohr oxidation is a major ATP source, breaking down fats to acetyl-CoA. 2, when this oxidation rate is very high in the liver, it produces ketone bodies via ketogenesis. 3, these ketone bodies are vital alternative fuels for other tissues, especially the brain during fasting. 4, both pathways are tightly regulated by FFA supply, by CPTI controlling mitochondrial entry, and by how acetyl-CoA is partitioned. And 5, problems in these pathways cause significant diseases from hypoglycemia to ketoacidosis. Understanding this is fundamental to metabolic health.
It absolutely is. This deep dive really drives home how interconnected everything is. Glucose metabolism, fat metabolism, energy balance. Seeing how the body adapts, switching between fuels is just remarkable.
It really is. And it leaves us with something to ponder, doesn't it? Thinking about the liver's central role in handling both glucose and fat, how might a chronic issue primarily affecting glucose metabolism, like the insulin resistance in type 2 diabetes, how might that ripple out and indirectly affect fatty acid oxidation and ketogenesis over the long term? What are the downstream consequences of those intertwined metabolic shifts throughout the body?
That is a fantastic question to reflect on. It really highlights the systemic nature of metabolism. Thank you so much for guiding us through all this detail. I feel like we've really achieved that deeper understanding we aimed for.
My pleasure. Biochemistry is endlessly fascinating, isn't it? The more you dig into these pathways, the more elegant the whole system appears. Hopefully everyone listening feels encouraged to keep exploring this incredible molecular world.